Okay, we should move on. So, the last talk of the session, last but definitely not least, is Morgan Levine, and from I think solely from Altos Labs. Is that right now, Morgan? Or
I still have an affiliation with Yale.
Okay, so Yale University and a founding member of Altos Labs in San Diego. So a great addition to the San Diego research community. And Morgan's going to talk about DNA methylation landscapes in aging and reprogramming.
So thank you very much, Peter, and thank you for Active Motif. I actually wore my Active Motif shirt today in honor of this conference. So yeah, you guys have heard already a lot about epigenetic clocks. I think they've been in at least 4 of the other talks. I'm going to talk about them, but really in the context of how we should think about them and how we should think about kind of using them as biomarkers of aging and what a change in the epigenetic clock might actually represent. So first, just this idea of biological aging. So epigenetic clocks or any of these other biomarkers were really developed with this idea of trying to quantify the biological aging process in contrast to the chronological aging process. But one consideration or perhaps even issue with this is the concept of biological aging is actually this latent feature. There's no ground truth and there's no actual observable biological age that a given organism has. And it's actually just this really complex multi-dimensional process. You can think of it almost akin to intelligence. There's no actual concrete way to measure it. And instead, we actually have to use different mathematical models to kind of approximate this in an abstract way. So similar to how this abstract painting gives you the idea that it's a picture of a face without directly being a face, this is kind of how we think about biological aging. Can we create something that's in the image of biological aging without the ability to actually measure it directly? So there we go. So epigenetic clocks are probably some of the most famous biomarkers of aging. And just quick background, even though people already covered this, is that they're based on DNA methylation. So this is methylation at these CpG dinucleotides, and there's 28 million of these throughout the human genome. However, most clocks comprise only about 100 to maybe a few thousand of these CpGs. And most of the epigenetic clocks were developed using something called supervised machine learning. So that's where you take some variable, typically this was chronological age, and then you take your DNA methylation data and you try to predict chronological age, or in the second generation clocks, things that we think are better indicators of biological age.
Morgan, can I interrupt for a moment? It looks
Yes.
like your slides are only showing like the upper left corner.
Oh,
They're zoomed really far in.
Yeah.
is that still weird?
Yeah.
Can you see them when they're like this?
Yes, they look full screen there. I
That's
don't know.
weird if they zoom all the way in. Hold on, let me try one more time. I don't want to take too much time. I can do it in this screen if it's not. Is that still zoomed in?
Yeah.
Same problem I'm seeing. I'm not sure why.
That too?
Yes.
Yeah.
Okay, I'm just going to go from here so as not to take time. It's probably not that big a
Yeah,
deal.
it should be fine unless you have a lot of we won't get the animations here. I don't know.
Yeah, I'll lose some of the animation, but I think the gist of it will come across.
Okay.
Okay, thank you for letting me know.
Yep.
So these epigenetic clocks have been developed for about the past decade, and in many ways are actually pretty decent biomarkers of aging. So they correlate with chronological age, as shown here on the left side. You can also take the residual, so kind of the difference between the predicted age and what you would expect for someone's chronological age. And we see that people who are predicted older tend to have increased risks of things like morbidity and mortality. So here it's showing survival probability for the bottom quartile, so who we think of the slowest agers versus the fastest agers. Interestingly, these same epigenetic clocks also seem to work in vitro. So you can basically grow cells in culture and you find that as these cells age in culture, you also see an increase in the epigenetic clock. And they seem to be responsive to some interventions, particularly epigenetic reprogramming using Yamanaka effectors or OSKM. But there's still some things that I think we need to consider when we're trying to think about whether these actually serve as good biomarkers of aging. So again, as I said, they do accomplish some of these things. They track with chronological age, although not perfectly, which we actually want, because we want something, a biological age that is distinct from chronological age. For the most part, they seem to be fairly good independent predictors of morbidity and mortality. And one thing that we haven't shown yet that the field needs to move towards is showing that changes in the clock also produce functional changes in some aging outcomes. So whether if you were to reduce the clock, would you actually also get a reduction in risk? And then the other issue that we actually need to still accomplish is have some mechanistic understanding of what these epigenetic clocks are actually quantifying. And then another thing that people often forget is this idea of reliability. So if you are to have a biomarker of aging, you want a biomarker that has minimal variation in kind of test, retest scores, and also limited batch effect. So for my talk, I'll first start with this reliability problem, and then I'll go towards how we're trying to develop some construct validity for epigenetic clocks and actually understand what they're measuring. So this is a paper that was recently published in Nature Aging by Albert Higgins Chen as the first author. And what we asked was if I have technical replicates, so I have basically one blood sample split into two, and I ran these epigenetic clocks on each of these samples, would I get the same answer? And of course, for a biomarker, you would hope that you did get the same answer. But what we actually found was that for most of the existing epigenetic clocks, we did not get the same answer. So the way to read these figures for these histograms on the x-axis is the difference between the two technical replicates. So you can see often you get 5 years, sometimes even 9 years difference between these technical replicates. And so we really thought, oh, maybe this is the end of epigenetic clocks for clinical trials. But in trying to solve this, we were actually able to develop a mathematical way to fix this issue, where instead of training clocks on individual CpGs, we were essentially training them on kind of higher order, higher dimensional signals. So the embeddings that you'd get from a PCA. And I won't go into the methods of that, but just to show that you can actually reduce the variation drastically between technical replicates in all of these clocks that we retrained. So why is that important? So it's important for a number of reasons. So one is if we actually want to longitudinally track people over time, looking at things like interventions. Originally with the clocks, you would see people kind of jumping all over the place in terms of their epigenetic age. And you're not sure whether these are true changes in epigenetic aging or representing noise. So what we actually found was when you cleaned up the signal, most of these kind of drastic changes were actually just due to noise, and people actually tended to stay pretty consistent over time. The other thing is if you're going to use these as an endophenotype in a clinical intervention, you actually want something with low noise because it gives you more power to detect effect sizes. So we actually ran power analyses for both two different types of data. So one, if you were to do this in a human clinical trial, or if you were to do high throughput screens in vitro, the difference in sample size that you would need using in gray is the original clocks and in blue are these new PC clocks. And what you can see is regardless of kind of the effect size that you expect to get from your intervention, this drastically reduces the number of samples or the number of people you need to enroll in your study. So that kind of gets at this idea of at least if you see a change in the epigenetic clock, we can feel a little bit more confident that it wasn't due to just random noise in these measures. But we still don't know what these changes actually represent. So what is kind of the underlying causality of these epigenetic clocks? And some people have even said, oh, well, it's akin to changing an odometer. You can change kind of this readout of the thing, and are you actually changing the underlying biology? I actually disagree that it's probably simply an odometer, but we still do need some data to actually link changes in epigenetic age to changes in function. And we also need work to actually decode epigenetic clocks and what they biologically represent. Excuse me. So one hypothesis that we have in thinking about what epigenetic clocks represent is that they don't actually represent a single thing, but that clocks are actually composites of multiple kind of epigenetic phenomenon that just kind of get captured in the same thing. So one analogy that I want to use, actually now being in San Diego and really getting to enjoy Mexican cuisine, is this idea that so in Mexican food, you source just a few types of local food. So often things like beans, rice, cheese, but you can make kind of this wide array of diverse entrees. And this is kind of how I think of the epigenetic clocks. So they're sourcing a few types of CpGs in terms of kind of regulatory features or what they biologically represent. And they kind of, each clock puts those together in a different way. And having more of one in one clock versus another might give you a different signal when you're applying that clock to an intervention or trying to understand mechanism. And really what we need to do is understand, quote, the ingredients that are going into the epigenetic clock, so the unique signals that are going in, what they represent. And then if you were to do an intervention or you were to apply these clocks, look at the distinct pieces along with the overall clock score, because you might see a change in the clock score, but it could be due to just one component of the epigenetic clock versus changing all the CpGs across the entire clock. So the way we went about trying to kind of deconstruct these and understanding the ingredients of these is to do clustering on CpGs. So what we did is we took all the CpGs that have ever been included, well, not ever, but in most of the famous epigenetic clocks, or about 6,000 of these unique CpGs. And we looked at them in a bunch of different data types, so different tissue samples, in vitro and in vivo. And we clustered them based on their patterns. So CpGs that change together. So when one increases, the other one also increases, or when one decreases, the other decreases. Oh, my animation's not going to work here. Uh-oh. Okay, I'll just do this. Okay. So the idea is that first you create these affinity matrices for each of these different data types, and then we combine them into a single one. And then each CpG gets assigned to a different cluster. So we're denoting these clusters based on color. And then what we can do is we can actually look at the contributions of each of these individual clusters to the overall clock score. And this is because actually epigenetic clocks are really simple in terms of how you calculate them. They're just linear combinations where every CpG is multiplied by a weight, and then you add it across. So what we did is we only summed the parts from each cluster. So we would take the blue cluster and just sum the CpGs in the blue cluster, green, yellow, and then of course, if you sum them all together, you get the full clock score. But now we can actually say what part of the state, how much signal is coming from each of these distinct clusters. And again, I have animation that's gonna mess this up. I'll just scroll through though. So first we just said, what are the distributions of these different clusters in the various clocks? And as you can see, they're pretty similar, at least across these 4 clocks. And there's 2 bars because first we looked at them in whole blood, and then we also looked at them in iPSCs because the weights could change a little bit based on the tissue or data type you're looking at. But then when we start looking at other unique clocks, which actually have the effect of chronological age completely taken out of them, you see fairly different distributions from what we saw before. So you get enrichment of these kind of CpGs that we assigned to this red cluster and some enrichment of this yellow cluster. We can then look at more what we call bespoke clocks. So the Jean clock is actually a mortality only clock, and you can see it's really compromised just 2 or composed of just 2 types of these CpGs. The hypo clock are these CpGs that tend to be hypermethylated but lose methylation with age in these WCGW regions. So they're in very low density CpG regions. And then the opposite is these epitope clocks, which tend to be these polycomb associated CpGs in promoter regions, CpG island promoter regions. And then lastly, we just asked the question, now that Steve Horvath with Illumina developed this new mammalian array, are specific clusters conserved, or are you missing entire clusters off the mammalian array? And what we found is actually most of these were represented in these conserved CpG regions as well. So one reason to look at the individual modules is in the context of interventions. So if I were to do an intervention and see a change in the clock, again, as I mentioned before, one would ask, is it happening across the entire clock as a whole, or is it really just driven by one type of CpG that's responsive to that intervention? And if it is just one type, you'd want to know what that CpG type actually represents. So first, I'm going to talk about one intervention that people have already mentioned today, which is just this epigenetic reprogramming phenomenon, where you can take a somatic cell, let's say skin fibroblast, and convert it back to what looks like an embryonic stem cell in terms of this induced pluripotent stem cell. So most epigenetic clocks show a reversal of the overall clock score. This is just one clock, but you can show it across almost all of the epigenetic clocks, where you have your original cell. After you apply OSKM for, let's say, a month, you get a complete reversion of the epigenetic clock. But we asked, again, is this universal? Is this happening across all the different types of CpGs? And what we found is that it's actually not, and it's really driven by just a few of these modules. So these are all the different clocks, and their overall scores are split up by the various modules. And you can see in terms of a change from baseline, which ones are the most responsive to this epigenetic reprogramming phenomenon over the course of these about 28 days. So overall, we find that these kind of green, yellow, green, and light blue ones tend to be the most consistently responsive to this epigenetic reprogramming phenomenon. The one interesting place was this Dunedin pace of aging clock, where this green, yellow one goes up. And what we actually found is that it's reverse coded in that clock. So what's called younger in all the other clocks is actually called older in the Dunedin clock. So then we asked, what do these three different modules actually represent? And this is a little hand wavy at this point. We haven't confirmed that functionally these are what they represent, but this is what it seems to be. So what we find is that the ones in this light blue module tend to be associated in these CpG islands, which makes sense because they were also enriched in the epitope clocks, which are these PRC2 associated clocks. The green tend to be in these shore regions, and then this green yellow tends to be in the open seas. We can also look at enrichment for binding of either transcription factors or histone modifications. And again, this green one really does seem to be these PRC2 bivalent promoter associated CpGs. The green one we think is something to do with maybe nucleosome occupied promoters. And then finally, this green yellow one seems to be some intergenic punitive enhancers because we see enrichment for things like EP300. So then we looked at the behavior of these three specific modules to try and get a better sense of what they actually represent. What we found is the three of them are very distinct. So in iPSCs, this green yellow one tends to be CpGs that are very hypermethylated. The green one tends to sit somewhere in the middle, so actually very heterogeneous in iPSC. So you can think of this as about half your cells are methylated in these regions and half aren't. And then the blue are very hypomethylated. And what we can see is with perhaps differentiation, you kind of get this flattening out of these. So the green, this one tends to flatten out. You get more kind of movement towards hypomethylation. This one flattens out. And then by the time you have aged, it seems fairly flat and not to show consistent signals. So we looked a little bit more at the age correlations in these three different types of CpGs when you look at the actual clock scores. So this shows these three are in brain, and I'm showing this for the PhenoAge clock because the PhenoAge clock was actually trained only in whole blood and only in adult samples, but we actually see really interesting patterns in other tissues and in non-adult samples. So this just shows kind of the log of age with the overall epigenetic score. And actually what this suggests is that most of the changes, or actually more rapid changes, are actually happening during the developmental time as opposed to during kind of aging in adulthood. And you can see this here where we kind of just restrict to this kind of developmental period versus the age period. This is in blood, in brain, blood, and liver. The other thing you'll notice is that the PRC2 associated one, this blue one, tends to have really strong age correlation, so much stronger even than the other two. And actually, what we find is it's almost consistent with the entire clock score in terms of the age correlations. So the other thing we looked at is aging in culture. So this idea that you could replicatively passage cells and actually see the epigenetic clock go up. So we asked within these individual cell modules, what's happening. So this is in astrocytes, and what we find is the blue one, again, goes up fairly linearly as a function of passaging, whereas the green one, and especially this green-yellow one, goes up and then comes back down again. And this kind of inflection point tends to coincide with when we start seeing increases in senescence in our population in terms of slowing of the doubling rate and also increases in beta-gal. And again, I'll remove my animation. We also looked at embryonic stem cells passaging. Interestingly, these first two actually seem to get, quote, younger as embryonic stem cells passage, whereas the blue one does show again to increase epigenic age as embryonic stem cells passage. And then finally, we also had age-term immortalized cells, where again, you see this very nice, clean increase in the blue, but some kind of perhaps selection that might be happening in the other two modules. We then also looked at blood cell subsets because a lot of the epigenetic clocks, particularly for intervention trials, are going to be assessed in whole blood. So it's important to understand how different blood cell types might actually be reflected in the epigenetic clock. And the way to read this is kind of the difference in epigenetic age in years between these different cell types. And what you can see is in this green-yellow one, you get much younger epigenetic ages for these naive B and T cells, whereas you get much older epigenetic ages for granulocytes. And in the blue one, it seems to be much older in things like memory B cells, which again might fit into this idea of replication driving these changes, given that memory B cells have much faster turnover rates. That being said, even though they show differences in these different cell types, we still see very high age correlations when we're looking at sorted cells in terms of the epigenetic pattern. So it's not just capturing a change in cell composition, but it's probably also capturing within cell type changes as well. And then finally, if you were to do an intervention and you see an effect on one module, you would actually want to know, is that a module that has some implications for health downstream? So then what we did is we looked at the effect or the association between these different modules and risk of mortality over about a 20-year span. This is showing all of the modules that we had, but I highlighted the three that I've been talking about. And what you can see is this green-yellow one seems to be highly associated with mortality risk. This is when we look at it in the PhenoAge clock and the Grim-Age clock, which are the two kind of most mortality associated ones. And I also did this both kind of without adjustment for cell composition, which is this one, and with adjustment, showing that it's not purely confounded by cell composition changes. Interestingly, this blue one, which again is this kind of replication-based one, doesn't in blood seem to show any association with remaining life expectancy or mortality risk. But that doesn't mean that it's functionally not important. And actually, we had a similar project that was going on in parallel where these same types of CpGs came up and showed some implications for risk of cancer when you're measuring it in that actual tissue. So I'm going to, excuse me, really quickly go through that. So basically, this is work done by my former student, Chris Mintier, where their idea was to actually train an epigenetic clock that was specifically capturing something about mitotic history or replication. So to do this, we actually use these H-chart immortalized cells because we actually wanted something that was, for the most part, senescent independent and track something that was not going to be slowing down. So Chris trained an epigenetic clock using H-chart immortalized cells where we trained it to predict population doubling. This is our training sample, our validation sample. It works really well. And then we applied these to different contexts. So we looked at just non-immortalized astrocytes, primary astrocytes in culture, dermal fibroblasts in culture, mammary fibroblasts in culture, and then different in vivo samples. So liver, brain during this development, adult brain, and skin. And again, you see this very consistent increase in this, what we're calling cell drift or replication-based epigenetic clock. And perhaps not surprisingly, these were implicated in these PRC2 regions. So these are similar to what the blue module was picking up. And these seem to be very important, what we found for cancer. So we looked at the difference between, these are different types of solid tumors. So the actual tumor versus non-tumor in that tissue, and we find an increase in epigenetic age for the tumor. We also looked at if you were to test the epigenetic clock in the tumor, if it was predictive of survival. And we find, although this is a very small sample size that needs to be replicated, we find some evidence that it's predictive of survival. And perhaps the more interesting thing was we looked at breast cancer, but we looked at the normal tissue. So women with breast cancer in their normal tissue, did they show an increase in epigenetic age relative to women's without breast cancer in their normal breast tissue? And we find some indication that there is an increase, which we think might suggest that there might be some epigenetic age remodeling that could precede or even sensitize tissue to tumorigenesis. Again, we need to prove this out, but that's where the evidence seems to point. And then the last thing we kind of looked at here was this idea. So a few years ago, Vogelstein and Tomasetti published this paper saying that there's this bad luck hypothesis that the risk of cancer is just a roll of the dice based on replication. So there's just a chance that you're gonna get some driver mutation with every time a cell divides. And they showed this picture where they showed lifetime risk of cancer in different tissue types plotted against total stem cell divisions. We looked at this in the context not of mutations, but actually epigenetic changes, where we looked at risk according to the SEER incidence rates across different tissue types, plotted against our epigenetic measure. And this is in normal tissue, non-cancer. And again, you find that tissue types that are more prone to cancer tend to be higher in this cell drift. These are from the same individuals, so there's not a difference in terms of chronological age. We also show that this tracks with the predicted turnover rate of these cells. And then finally, again, just to retouch on the reprogramming thing, this replication-based epigenetic clock does seem responsive to OSKM reprogramming, where you get mostly during the maturation phase. So this is the dedifferentiation phase of reprogramming, a rapid decline in the epigenetic clock. And then we noticed this slight uptick. So we actually looked at more longer term passaging of embryonic stem cells, again, in iPSCs. And we do see that even though it reaches this kind of bottom point, as you start passaging these cells again, it starts ticking up again. So in conclusion, epigenetic clocks do meet some criteria for good biomarkers of aging. They have some construct validity in terms of being able to predict things other than age, like morbidity and mortality. And we think that different statistical applications like the PC clock method that we showed to improve reliability can actually help them be better biomarkers down the road. At the same time, they're not a singular phenomenon. So when you look at a change in the epigenetic clock, you need to actually think what that means and why you got that change. So even though reprogramming seems to reset the overall epigenetic clock, that really came down to just 3 types of CpGs that were in the clock. So these PRC2 bivalent associated ones, which are blue, that show an increase in methylation with age. We think that they're replication driven, and they show very strong, some of the strongest age correlations across tissues. But when measured in blood, they have weak mortality prediction, but perhaps are implicated in tumorigenesis. Then these kind of green-yellow ones, which we're thinking are probably associated with intergenic putative enhancers. They start hypermethylated, but lose methylation with age. There's maybe selection or some interesting kind of switching that goes on with senescence. So the epigenetic age increases up until senescence when you start seeing this kind of reversal. They seem to be associated with T-cell differentiation and their strong mortality signal, but this is even independent of the cell composition. And then these nucleosome occupied promoters, which are the green, also are hypomethylated or become hypomethylated with age. They're somewhat heterogeneous in iPSCs where they were at that kind of 50%, and they have some mortality association. So with that, I want to thank my lab at Altos, also Dr. Higgins-Chen, who kind of led the PC clock development, Chris Mintier, who worked on the replication-driven PRC2, and the other collaborators for that project. And happy to take any questions.
Great talk, Morgan. Fantastic. So yeah, we have a bunch of questions coming through. I'll start if you don't mind. I mean, I was interested in the technical what you described as noise. I mean, presumably that's technical variation between measurements, and you addressed it through this principal component method, which I don't think I really understood. But I wonder if it's technical, then surely is there a way just to address it by figuring out, you know, whereabouts in the measurement the technical variation is coming from, and just eliminating it technically?
Yeah, so that's a great question. That was actually our first approach when we tried to deal with this, was that maybe there are just a few CpGs that are just prone to noise in the clocks. And what we actually found was each sample, the technical noise is coming from a different CpG. And there are some that are a little more prone, but throwing them out actually made the clocks worse in terms of their prediction of aging outcomes. So those CpGs are still really important. So what we did is the statistical way is you're almost averaging across CpGs that have the same signal. You're kind of averaging the signal from them with the idea that technical noise in one will actually not have a big overall effect on the clock, versus just, you know, taking a kind of one CpG as representing that whole signal, which then is prone to noise because it's kind of, you're focusing all of the signal on this one variable.
I see. But is the technical is the noise then is actually coming from the Illumina arrays that you're using? Something
we're not sure exactly which kind of step the technical noise kind of creeps in, but it's the amount of noise is fairly consistent across the different CpGs on the array, and then the ones that just have smaller kind of ranges tend to be more prone to that because that absolute amount is bigger compared to their standard deviation.
Right. So Laurie Boyer asks, says great talk. I mean, have you compared your data with single cell transcriptomics, and in particular, you know, looking at the different kind of categories of clock that you've deconvoluted? Is there any relationship between the different types of clock, and and changes in gene expression within those groups?
Yeah, so we do have single cell RNA-seq data for the serially passive cells that we're analyzing now. But we're also collecting a bunch of other kind of multi-omic data to try and kind of layer this on. A lot of it will be bulk, some of it will be single cell. One issue is that I think Vadim talked a little bit about this, is that single cell methylation is still in its infancy. So actually understanding how much of these changes are coming from population shifts versus changes within cells is still not clear.
Right. Yeah, and talking of Vadim, Valentin Krakan mentions Vadim in the context of metabolic clocks. So Valentin, wants to know, well, first of all, is it possible to develop a metabolic clock? I think there is some evidence for such a thing, but so I guess more generally, what's your thoughts on the pros and cons of different types of potential clock, metabolic clock, transcriptomic clocks? I think now there's even a brainwave clock that whereby you can measure people's brain waves in their sleep, and that can also be used to measure chronological age. So what do you know about the pros and cons of different types of clock?
Yeah, I mean, there are so many clocks coming out, and I think that just shows so many things change with age. So you can almost take anything and develop an aging clock out of it, because there's hardly anything that stays kind of invariant to age. And I think what we'll find as we develop more and more of these is they're probably capturing different signals. They'll have some overlapping signal, and that might be where kind of the meat is, where you want to kind of measure aging, but they'll also have distinct features that each one can individually capture. The one thing that made me so fascinated about methylation clocks versus other things is their universality. So you can use the same clock across different tissue types. You can use the same clock in vitro, and now Steve Horvath showing across mammals, which I don't think has really been shown for any other type of clock, but we might find that down the road for other ones as they're explored a little bit more.
Right. And I know you're doing quite a lot to try and extract, I think, from blood, but use measurements in blood to measure the biological age of different tissues and kind of organ systems. So I wonder whether you think that, different types of clock, I mean, maybe a brainwave clock might be particularly good at measuring neurodegeneration, for example. Do you think that's likely?
Yeah, absolutely. So I think the problem is we talk about biological age, like everyone has one biological age similar to one chronological age, but really all our body parts, even all our cells, are aging at different rates. And you might want a different clock depending on what organ or tissue you're profiling. We are developing this system age clock, which uses blood to try and capture the different organs, because you're not going to take biopsies of
right. Absolutely. Yeah. So I think, I totally agree. I think that, different, even within the same person, different tissues probably have different biological ages. But yeah, so if we can measure those, those different tissues in blood, that's obviously a very attractive thing to do. Zhizhen Do has a question. Go ahead.
Okay. So I have a question about the commercialization of the epigenetic clocks, because apparently there are many companies working on it. And I also became very interested in knowing my own epigenetic age. Well, one thing is that I see it's not cheap. But the other thing is that I guess my bigger question is that, say, if the clock tells me I'm 20 years or I'm 80 years old, right? So what exactly can I learn from this? So the question is, can you comment on how the epigenetic clock, the age, is going to benefit the health of the general public, the healthy individuals? And a related question is, can you comment on where we are and what do you think is the future promises for this industry?
Yeah, I think those are kind of two related things. I think we're still very, very early. So I would caution anyone who's going to put too much faith on their measure they get back. You can do it for fun. And I think, you should do it. And if you're going to think to actually evaluate that, you should do it in the context of lots of other things too. Get a normal blood panel or physical done and if everything agrees that you're doing pretty well or not doing well, then I think maybe trust it. But I would never just trust the clock score you get back by itself, independent of everything else. The other thing to keep in mind is every company is going to give you a different age. So how do you interpret that? Because they all use completely different clocks and different reference populations to get that age. More than the actual age you get back, I think, again, assuming they're reducing this technical noise is kind of your change over time. So I would be more interested in from year to year, whether I was increasing a whole year or less than a year. But like you said, they're really expensive at this point. So, it's not something that I'm going to suggest everyone go out and spend $500 to get their epigenetic age measured every year. I do think they have a lot of promise. So, we don't want to throw the baby out with the bathwater. They're not ready yet, but I do think the field can keep moving as we understand them a bit better and understand their applications and their, the caveats. I think they will have a lot of promise, probably in more of the medical context than, the direct to consumer, who knows? But yeah, I think there's still a lot we can learn.
Thank you.
Okay, so 2 or 3 more questions for Morgan, and then we'll transition into the round table stage. So Gary has a question.
Thanks, Peter. So Morgan, I actually have 2 questions. I may hold one for later. But you mentioned in one of your earlier slides that you were looking at cancer patients. I was wondering if you'd looked at patients that had undergone chemotherapy to see what that looks like over time, because you could see how that would have an application, right, in medicine and in health.
Yeah, so I was part of a collaboration where they looked at radiation therapy, and they did see increases in epigenetic age in association with that. And I think there was reduction if they also did an exercise intervention. But I would imagine, again, that chemotherapy will show an increase in epigenetic age, but I haven't looked at that specifically in my work.
Right, because my thoughts were that then you could take treatment measures to ameliorate the impacts of whatever type of chemotherapy that you would give to a particular patient. So, yeah, thank you.
Ognian, go ahead.
Hi, thank you for the fascinating talk. I think I'll be thinking about and processing the information for next week at least, if not more. If I can add my very naive contribution to the debate on the noise of epigenetic clock, and also follow up with a question about that. So my personal thought and understanding is that this is the problem of noise is a little bit intrinsic in the way that the clocks operate, because they're, in a way, they're very fragile models. They're very sensitive. So they're linear models. So each locus is additively added to the sum total. So, and some of the so we multiply the percent methylation by the coefficient, and some of these coefficients sometimes are quite large. So even one mistaken or problematic locus for whatever reason can add several years to the estimate. And of course, on average, these will cancel out, but they don't always cancel exactly. So from that point of view, also, I would expect, I don't know if that's the case, and please correct me if I say nonsense, I would expect that clocks that have more loci should have higher noise, because if this is the problem, then the more loci we have and the higher the coefficients are, the larger the noise should be. I don't know if that's the case. And from that point of view, I think I find it really exciting, the development of the principal component clock, because this is potentially a very good way to get rid of that noise, because what principal components do really well is that they capture the main trends in your data and they ignore all the noise that you're not really interested in. So I wanted to ask you a bit more about how you calculate your plot, because I have to admit I read both the preprint and the published paper, and I still haven't figured out exactly. So you have your data point, which is based on the linear model. Then you calculate presumably principal component. You detect the first principal component, which would be the main trend, and you project on that principal component. Is that how it works?
No, so you don't take the linear model for the clock at all. We ignore the original models for the clocks completely. You take your methylation data, so your matrix of cpgs by samples, and then you calculate, you get all the principal components. So we put out all the loadings for them. So you get your PC matrix from that. Then each of the PCs that were selected have some weight associated, and you multiply them by a weight and then add, but it's an entirely new equation. It's not, it actually doesn't use the original clock equations at all. We went back to the initial data and retrained them all, but yeah, the original weights are not used in the PC.
Okay. So you start from the matrix of DNA methylation levels itself, and that's your
and then you get
hyperspace.
your eigenvalue matrix, and then the eigenvalues get multiplied by weights and then summed. And we usually have about 500, a few hundred PCs end up contributing. So it's not just PC1 that contributes to your clock.
Oh, okay, I see. So if that's the case, then what makes the PC Horvath clock be a Horvath clock and the PC PhenoAge clock a PhenoAge? Do you use the same data sets? Is that?
Yeah, for PhenoAge, we updated it because the original data set was a little bit small. So we combined it with a bigger one. The Horvath one, we didn't have access to all the samples that Steve originally trained on, but we tried to get surrogates and then show that we, weren't losing anything in terms of construct validity in the new one.
Okay, that's great. Thank you.